Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we were discussing about cancer biology, in which we already discussed about hallmarks of cancer, and we have already listed down all of the hallmarks of cancer, and we discussed about the two very important hallmarks in the last lecture, the how the tumor cells they are able to sustain a proliferative signal and how they are able to evade growth suppression. So in that lecture, we learnt about proto-oncogenes and tumor suppressor genes. So let's summarize the lecture. So we learned that there are a set of genes known as proto-oncogenes which are very important for the growth. So they are very important for the cell differentiation and cell proliferation. So these are generally the growth factor receptors, or some signal transducer that are going to signal for the growth of the cell. So those proto-oncogenes, when they undergo gain of function mutation, okay, they are converted into the oncogenes, which are going to lead to the cancer. Okay, so this is going to lead to the cancer. So this gain of function mutation in proto-oncogenes, this is usually a dominant mutation. Then we also learned about the tumor suppressor genes. When those genes undergo loss of function mutation, they upon losing their function of suppressing the tumor, so they lead to the uncontrollable growth of the cell, which leads to the cancer. Okay, and generally those tumor suppressor gene, the loss of function mutation of tumor suppressor gene, that follows two hit hypothesis, means there should be two recessive alleles to the accumulation of two alleles is quite important for a tumor suppressor gene to result into the cancer. So this is what we learned in the last class. In today's class, we are going to learn about a very important example that is listed down in almost all of the books about cancer biology. And so this example is very important. So we are going to learn about a pathway which, if disrupted, leads to the cancers. Today, we are going to learn about retinoblastoma and E2F interplay, which if disrupted leads to the cancer. Okay, so let's take a look at the scenario when a cell it is at the rest, or let's say that a cell is in G1 phase. Okay, so a cell generally follows this fashion. The life cycle of cell, lifespan of the cell is something like that. First it has a phase known as growth one phase, where it grows and prepares for prepare for the division. Then it enters into the S phase, where the DNA replication happens, and then it enters into the G2 phase, the preparatory phase for the division, and then it enters into the M phase. M phase is the division where is the phase where the actual cell division happens. So this is a general overview of the cell cycle. Okay. So in this cell cycle, there are certain checkpoints which regulates the transition from one phase to other phase. So the retinoblastoma, this protein, is involved in the transition of a cell from G1 to S phase. So the G1 to S phase checkpoint, in this checkpoint, retinoblastoma, which is abbreviated as RP, this protein is going to play a role. Okay, so what happens is that when a cell is at G1 phase, okay, when cell is at a G1 phase, there's a transcription factor known as E2F, and this transcription factor is very important for transcription of the genes which are involved in DNA replication. Okay, so to this transcription factor E2F, there's a protein which is bound to that in the G1 phase, and that protein is known as retinoblastoma. Okay, so this complex, the RB and E2F complex, is present in the cell during the G1 phase. So what happens is that there will be certain mitogenic signals which are signaling. So there will be some growth factors which are going to signal that now the cell needs to undergo to the division. Okay, so now the cell is ready for undergoing the division. So now the cell should progress from G1 phase to S phase. Okay, and those upon the arrival of such mitogenic signals, the signals which are responsible for mitosis, okay, the division, cell division. So upon arrival of such signals, the retinoblastoma protein is phosphorylated. Okay, so this, okay, yeah, I drew it in blue, so let's change the color. So this retinoblastoma protein, this is phosphorylated. Okay. And due to the phosphorylation of retinoblastoma, it undergoes some conformational changes, which breaks down the bond, so which breaks down the bond between the E2F and retinoblastoma. So now, due to the phosphorylation of retinoblastoma, E2F transcription factor is free. So what happens is that upon the arrival of mitogenic signals, retinoblastoma gets phosphorylated, which results into the E2F release. Okay, so now this E2F being a transcription factor, it is going to control the transcription of certain genes. So now what happens is that this is a DNA, okay, and when once this retinoblastoma and E2F complex break down, this E2F goes and binds to the DNA, and now it facilitates the transcription of the genes which are very important and crucial in the DNA replication. So now once the so these genes are they are transcribed, that will lead that will lead to the DNA replication, means the cell has progressed from G1 phase to S phase. So this is a general overview of what happens in a cell from the transition of G1, from when it transits when the transition happens from G1 phase to S phase. So this is a normal scenario. So this is a very tightly regulated process, and if this process gets disrupted, it is going to lead to the cancer. Now I'll leave this up to you, that what dysregulation should be there for this certain pathway to lead to the cancer. Just pause the video and think about it. Okay, so the cancer means the uncontrollable growth of cells or uncontrollable proliferation of cells, right? So for uncontrollable proliferation of, for the cell to go from G1 phase to S phase, what do you need? First you need the free E2F, right? So you need to release E2F from the retinoblastoma. So when this E2F release, it is timely and it is in control manner. It is not going to lead to cancer. But when this gets uncontrollable, so if there is constitutive activation of E2F, means E2F is free all the time, that will lead to the cancer, that will lead to a normal cell, transition of a normal cell to a tumor cell, right? Or something goes wrong with the retinoblastoma and it always stays phosphorylated. So let's say that there is a mutation in RB gene. Which kind of mutation in RB gene will lead to the cancer? The loss of function mutation. How? So what is the function of retinoblastoma protein? Retinoblastoma protein is going to hold down E2F and it is going to inhibit E2F. It is going to, it is going to hold back the E2F and it is not letting it to transcribe the genes which are required for DNA replication. So this retinoblastoma is a tumor suppressor gene. Why? Because if this gene, due to this gene, there is a control on the cell proliferation. So if the gene is absent, if there is no retinoblastoma, the E2F will always be activated. So E2F is always free and it is going to always transcribe the genes which are important for DNA replication. And the DNA replication will go on, which will lead to the uncontrollable proliferation, which in turn leads to the cancer. So that is why the loss of function, if the retinoblastoma protein, if either retinoblastoma protein is not produced due to the mutation in RB gene, or if even if it is produced, but there is some some defect in that protein itself, so that it cannot hold back E2F. So the loss of function mutation in RB gene, that is going to cause the cancer. Okay, so these are two important things. These are two important events that are going to lead to the cancer. But there's one more point which can lead to the cancer, and that is the mitogenic signals. So here, when I discussed about mitogenic signals, actually there are two signals, there are two factors, two CDKs that are going to lead, that are going to make this transition from G1 to S possible. The first complex is known as CDK4/6 and cycline D complex, while the second is CDK2 cycline E complex. So both of them are very important for the transition of G1 phase to S phase. So these are they so CDK4, 6 and CDK4 or 6 or and cyclin D. This complex, this initiates phosphorylation. This initiates phosphorylation of retinoblastoma protein, while the CDK2 and cyclin E, they they take the phosphorylation to a further level and activate E2F. So both of them, both of those complexes are important. So they kind of serve as a signal for the cell that it has to move on from G1 phase to S phase. Okay. So what if those gets upregulated? So if there due to some changes in the gene which are going to produce cyclines, okay, which are produced in the CDK, if the CDK4/6 or cyclin D, or let's say CDK2 and cyclin E, if those factors gets upregulated, there is overexpression of those genes, that is also going to lead to cancer, because they will serve as a continuous signal for the proliferation. So now they will serve as a continuous signal for the cell that it has to undergo the transition from G1 phase to S phase. Now the cell will undergo into the cell will keep continuously keep replicating the DNA, and that will lead to the cancer. So those three things, they are going to, so if the E2F release, if there is constitutive activation of E2F, or if there is loss of function of retinoblastoma gene, or if there is overexpression of the CDKs, all of those three, they are going to lead to the cancer. So what could be an escape route for them to, for cells, for not causing the cancer? So for avoiding the constitutive activation of E2F, what we can do, we can either, we can either introduce the E2F inhibitors, or the inhibitors targeting E2F targets. If if either of them if either of them is achieved, that is going to lead to the inhibition of transition of a normal cell to a cancerous cell. Because if the E2F inhibitors are present in the cell, even if the E2F is released from the retinoblastoma protein, now this E2F will be inhibited, and there will be no transcription of the genes which are required for the DNA replication. And this is how the normal cell will not be converted into tumor cell. Okay, so this can be an escape route for a cell, for not getting converted into a tumor cell. The second, so for the if the retinoblastoma gene is mutated, if it loses its function, what we can do? We can either introduce functional retinoblastoma protein, or we can even introduce functional retinoblastoma gene. These are the hypothetical escape routes for a cell to undergo for inhibiting the conversion of a normal cell to a tumor cell. Okay, or we can even introduce the inhibitors, the CDK inhibitors in the cell so that the cell do not undergo, so the cell do not form the tumor. Inhibitors of CDKs, they can also, they can also help us to stop the cancer cell progression. So this is it in for today's class. In next class we will learn about other pathways, the RAS pathway, which are going to lead to the cancer. RAS pathway, the BRAF mutations. Till then, see you, bye bye.